In this video, we're going to talk about getting to translation and specifically the genetic code. And so where we're moving now with our if the unit is we're moving on to the next step of the expression of our genome or gene expression. We talked a lot about the mechanism of transcription, of turning a gene into RNA, so transcribing a gene into messenger RNA. We talked a lot about the regulation of this process, and now we're going to look at the next step of gene expression, which is to translate that messenger RNA into protein. And then, of course, that protein is going to fold such that it has a specific structure, which gives it a specific function, like hormones and transporters and enzymes, which will then give that cell in which those proteins are expressed a specific structure and a specific function. That's called cell differentiation. So abstractly, we can say what we're looking at is we are looking at the mechanism by which the genotype, all the information within your genes, is converted into your phenotype, all of the structure and function of all of your cells and giving rise to all of your traits. So we need to first look at the genetic code, which is the code necessary to convert the information in nucleic acids, specifically messenger RNA, into proteins, specifically amino acids. And this is the genetic code. Now, I first want to say that this table that you see here will be on your exam. So you don't have to memorize anything, but don't get complacent. Don't take this for granted. You still need to learn how to use it. The way you use it is, first of all, each one of these three letters is a triplet codon as it would exist in the messenger RNA, reading it from 5' to 3'. And so each one of these codons, the first letter is 5', the last letter is in the 3' position. Okay? And what you'll immediately notice is a triplet codon, okay, gives rise to 64 possible codons. Now, I want you to do that math. How could you do the math to derive this 64 possible codons? I'll give you some time to think about it, and we'll talk about it in lecture. Now, there's only 20 amino acids, right? Because each one of these codons is going to encode an amino acid. And then there's these 3 stop codons that do not encode an amino acid. So there are 41 extra codons, 41 codons that we don't need, that the cell doesn't need, right? 23 is plenty. So we need to figure out what it is the cell's doing with those 41 extra codons. Well, how is a codon read? What reads a codon? Well, the answer is that the anticodon of a transfer RNA that carries a specific amino acid, and we'll get to that in a later lecture, is what binds to the codon of a messenger RNA, and it does so in a reverse complementary fashion. So if the codon is 5' AUC, the anticodon that binds to it is 3' UAG. Okay? So you've got this codon-anticodon relationship. So I've got this question here. What amino acid should be connected to the tRNA? And here's the thing, folks. To figure out which amino acid should be connected to this tRNA, you don't go and look and say, oh, it must be the GAU. It's not the anticodon that's written here within our genetic code. It's the codon. Okay, just a reminder. So we look at AUC. We find that here. So AUC, so it's isoleucine, should be connected to this tRNA because it has an anticodon that recognizes AUC, which is the codon for isoleucine. Okay, so that's how that works. Now, we have already mentioned this, that there are 41 extra codons that aren't necessary. And what we see then is what happens to those extra codons is they're just assigned to different amino acids, such that some amino acids are encoded by not just one codon, but up to 6 codons. Arginine, leucine, serine, all of them are encoded by 6 codons. There are a few methionine and tryptophan that are only encoded by one codon, but most of them are encoded by more than one. This is called redundancy or degeneracy. The genetic code is such that there are several amino acids that are encoded by more than one codon. Now, what you are going to have to pay attention to is what are the molecular mechanisms by which this occurs, by which a single amino acid can be encoded by 6 different codons. Now, there's one fairly simple way in which redundancy is achieved, but there's also a more complex way that we'll look a little bit later. So the first is called the wobble concept, the wobble effect. And the wobble effect is an effect of the structure of transfer RNA. So here is a transfer RNA. You can see it's just one RNA molecule that's folded up on itself quite a bit there. And right here, right at this corner, this fold, is our 3 base pair, our 3 base anticodon, such that the 5' most position comes right at this bend, right at this turn. That is the wobble codon, or I should say wobble anti-base, okay, anticodon base. And because it is in this position, it can wobble, it can flex, such that it can accommodate some base pairing that is not always possible. What do I mean by that? Now, if in that 5' position, that wobble position, if it's a C or if it's an A within that transfer RNA, there's nothing special that happens. A C is only going to bind to the G in the codon. The A is only going to bind to the U in the codon. Now, if, however, that 5' position within the anticodon is a U or a G, the fact that it has this wobbling that can occur here at this turn allows it to not only base pair with an A, but also a G. This G can not only base pair with a C, but also a U. And this means that a single anticodon that's part of a single transfer RNA that holds a single type of amino acid can go to 2 different codons, the XYC codon and the XYU codon. And there you have it. There is a way in which 2 different codons encode the same amino acid. Next, post-transcriptionally, some tRNAs are modified such that what was originally an adenine in this 5' most position of the anticodon is converted, is chemically modified to something called an inosine. And an inosine can hydrogen bond with a uracil, a cytosine, and an adenine. A cytosine, a uracil, and an adenine. So now we have a single, so if any of these transfer RNAs contain an inosine in that 5' most position of the anticodon, it can bind to 3 different codons, which means that amino acid that's carried by this transfer RNA is encoded by 3 different codons. So between the wobble effect and this 5' inosine, those are 2 of the 3 ways in which redundancy is achieved within the genetic code. Now, the third way we're going to look at in another lecture. Let's take a look at the rules of the genetic code. Number 1, it's non-overlapping, meaning every base within a messenger RNA only is in one codon. So this A is only in codon 1, this U is only in codon 1, this A is only in codon 1. Now that's to contrast it with an overlapping code in which some bases would be in more than one codon, right? This A here is in codon 1, it's in codon 2, and it's in codon 3 because of this overlapping code. Okay, but that's not the case. The genetic code is non-overlapping. All right, there's only one proper reading frame within the messenger RNA, and it's not necessarily the first reading frame. So I've got some important questions to ask ourselves as we're kind of talking about this. Well then, what determines which of the 3 reading frames is read? What determines where the reading frame starts and stops? And what is before and after that start and stop? So let's take a look here. We have 3 possible reading frames. We start here, ACG, UAU, right? That's one reading frame. And then if we shift and start one over, we have CGU, AUG, GUC. And then if we shift 2 over, 1, 2, and now start here, we've got GUA, UGG, and so on and so forth. If we then try to go into a 4th reading frame, so we shift over 1, 2, 3, and start here, look at that. UAU, we're right back into reading frame 1. Okay, so a codon that's made up of 3 bases has 3 possible reading frames, only one of which is right. Which one is that? Well, what happens is the ribosome binds the 5' end and it scans all 3 frames until it hits the first AUG. In whatever reading frame it can find it, that's it. So that first AUG, starting with the 5' end, that locks it into, this is where the reading, this is which reading frame we're going to be using for this messenger RNA. And number 2, this is where we're going to start reading. The AUG is the start codon. Which means all of this stuff upstream of that first AUG, all of this stuff does not encode protein. It's called the 5' untranslated region. So if we take a look at a messenger RNA in which if we start at the 5' end and we go in all 3 reading frames, right here is the first AUG. So all of this what, 10, 20, 30, 40, 50 base pairs, that is the 5' untranslated region. It's an exon, right? It came from an exon from that gene. It's not an intron, so please don't get confused with that. It made it to the cytoplasm, but it doesn't encode protein. It has some important purposes, and we will get to that, but it does not encode protein. Now, we go through in that reading frame dictated by this first AUG, go through. All of this is what's called the open reading frame. This all does encode protein until you get to the stop codon. Boom, stop. This is where the ribosome drops off. This is the protein is finished. Now look at all of this. This massive 3' untranslated region. None of this encodes protein because the stop codon was right there. Okay. Again, this was part of an exon. This isn't like it was an intron that made it to the cytoplasm. This is all part of an exon that was spliced together with all the other exons. But it doesn't encode protein. It does have important role to play in the stabilization of messenger RNA and the regulation of messenger RNA, and that we will get to later. Now, the genetic code is universal. Humans, E. coli, plants, amphibian, viruses all use the same genetic code. There are a couple very, very minor exceptions, okay? But they are, for the most part, absolutely universal, which means you can take a human gene, pop it into bacteria, and that bacteria will read that gene, write the messenger RNA, translate that messenger RNA just fine, and that's absolutely essential for recombinant DNA technology. All right, so the next lecture, we're going to look at the components that are necessary for translation, okay? And then in the lecture after that, we'll talk about the mechanisms of translation.